ChimeraX is a powerful next-generation molecular visualization tool developed by the UCSF Resource for Biocomputing, Visualization, and Informatics. Designed as the successor to UCSF Chimera, ChimeraX offers advanced capabilities for interactive visualization and analysis of molecular structures, density maps, trajectories, and more. Its modern graphics engine allows for high-quality rendering, making it suitable for both publication-quality images and exploratory research. The software supports a wide range of file formats commonly used in structural biology, such as PDB, MMCF, and cryo-EM maps, and can handle very large datasets efficiently. ChimeraX also provides tools for structure editing, molecular docking, sequence alignment, and scripting through Python, enabling automation and customization of workflows. A key feature of ChimeraX is its user-friendly interface, which includes both a graphical interface and a command line for advanced users. It is compatible with Windows, macOS, and Linux, and is continuously updated with new features and improvements based on community feedback. ChimeraX is widely used by researchers, educators, and students for tasks ranging from visualizing protein-ligand interactions to preparing figures for scientific publications. Its extensibility and integration with other bioinformatics tools make it a valuable resource for molecular modeling and structural analysis.